Hello everyone, welcome to the Tim Booker channel. I hope you enjoy listening to books. Today, the book I'll be interpreting for you is The Incredible Skeleton. Humans walk on two legs, birds soar with wings, hermit crabs dwell in shells, and insects defend themselves with exoskeletons. Behind these natural phenomena lies a common physiological structure, the skeleton. Undoubtedly, the importance of the skeleton to life cannot be overstated. It serves as the framework supporting an organism's body, evolving into the most practical tool throughout biological evolution. In a sense, the appearance of the skeleton even completely altered the course of life's evolution. Throughout the Earth's approximately 4 billion years of existence, for most of that time, life's evolution remained rather primitive. If one could travel back to those times, the surroundings would display minimal traces of life. However, about 500 million years ago, the Earth suddenly teemed with vitality. Diverse life forms rapidly emerged within a short period, marking a new chapter in life's evolution. One significant reason for this shift was the advent of the skeleton. Hence, to comprehend nature, the skeleton stands as an unavoidable topic. Traditionally defined, the skeleton generally refers to the hard tissue within or on the surface of an animal's body. Yet, the author of this book presents a broader concept of the skeleton. For instance, tree branches constitute the skeletal structure of plants, while the glasshouses of diatoms serve as the skeletons of microorganisms. Through this book, we learn that the skeleton is a product of an arms race among different species. Its appearance in The Incredible Skeleton further ignited a broader competition, thereby hastening the evolution of complex life. Over the span of 500 million years, various species developed their own types of skeletons, leaving behind invaluable information within these skeletal structures for scientists to unearth secrets from the past. The evolution of the skeleton falls within the realm of paleontology. Both authors of this book are renowned scholars in this field. The first author, Jan Zalasiewicz, is a British geologist and paleontologist, an honorary professor at the University of Leicester, and has served as the chair of the International Commission on Stratigraphy's Working Group on the Anthropocene. The second author, Mark Williams, is a British paleontologist and serves as a professor of paleontology at the University of Leicester. Now, let us embark, guided by these two paleontologists, on an exploration of the incredible skeletons found in nature through 3 sections. 1.in the first section, let's discuss when the skeleton appeared and why organisms evolved to have skeletons. 2.in the second section, we'll continue exploring how the diverse skeletons in nature evolved. 3. Finally, in the last section, we'll delve into what past secrets we can uncover through the study of skeletons. Let's start by discussing when the skeleton first appeared and why organisms evolved to have skeletons. The great biologist Charles Darwin noticed an intriguing phenomenon early on. In rocks dating from the Cambrian period onwards, there was an abundance of ancient fossils. However, in rocks older than the Cambrian period, there were scarcely any signs of life. Darwin hoped to observe older fossils but instead encountered what seemed like a world devoid of life. The Cambrian period appeared to be a demarcation line, before it, silence, after it, vibrant life. What could be the reason for this sudden shift? You might have guessed, it's related to skeletons. The ancient biological fossils we see today mainly consist of skeletal remains because the soft tissue structures of organisms are challenging to preserve during fossilization. So, before the appearance of skeletons, although life had existed on Earth for over 3 billion years, these life forms were either minute single-celled organisms requiring a microscope to observe or simple soft-bodied creatures lacking skeletons, making fossilization difficult. Therefore, when Darwin examined older rock layers before the emergence of skeletons, he couldn't detect traces of life. But when did the earliest skeletons appear? Currently, the earliest known animals possessing skeletons were the Cloudina, named after American geologist Preston Cloud. Cloudina had irregularly shaped curved tubes, quite small in size, mere millimeters wide and a few centimeters long. They first emerged approximately 550 million years ago, very close to the Cambrian period. Scientists analyzed them and found that the tube-like structure of Cloudina was likely composed of calcite, fundamentally calcium carbonate. As seawater was rich in carbonate and calcium ions, Clodina's skeletons easily utilized these ions from the seawater through certain biochemical mechanisms to form their skeletons. However, why did Clodina pioneer and invent this unprecedented structure, the skeleton? The appearance of another type of fossil might provide an explanation. Sometimes, traces of ancient biological activities left behind can also fossilize. In rock layers contemporaneous with Clodina, there exist numerous unprecedented trace fossils, burrow fossils. Burrow fossils record imprints left by ancient organisms as they traversed or stirred up sediments on the seafloor. The presence of these fossils implies that some organisms had acquired the ability to tunnel at that time. What did this ability signify? Among well-preserved Clodina fossils, about 1/5 showed signs resembling drill hole attacks, such as circular perforations on their shells. Hence, scientists speculated that Clodina's skeletons likely functioned as armor, protecting them from predators. With an offensive spear comes defensive shield. The emergence of the skeleton marked the opening of a new chapter in the arms race among organisms. Shortly after Clodina's appearance, around 540 million years ago, the famous Cambrian explosion occurred. Ancestral forms of most present-day animals rapidly emerged in a short period, transforming a once silent world into a thriving one, much like what Darwin observed. Subsequently, creatures with skeletons quickly expanded from just a few species to an uncountable multitude. The renowned trilobites chose skeletons for protection, while the apex predators of that time, anomalocaridids, utilized their robust skeletons as hunting weapons. Additionally, numerous unknown species left behind varied fossils after death, collectively termed small shell fossils. These encompass various minute button-like, tube-like, and shell-like fossils, each merely a few millimeters in size. Though distinguishing these fossils' origins today might be challenging, it's certain that they represent a vast array of species. It's evident that since the Cambrian period, skeletons rapidly became prevalent among different species. Reflecting on this history, it's apparent that initially, skeletons might have served as armor for some creatures, but swiftly became a crucial tool aiding survival in intense competition. This rapid proliferation is understandable. To defeat well-equipped adversaries, the best strategy is to be better equipped. Because of the monumental impact of skeletons on geological strata, in a way, even the Cambrian period itself is defined by skeletons. The era of skeletons began then and has persisted until today. During a period before and after the Cambrian explosion, the evolution of skeletons underwent a significant transformation from non-existence to existence. Subsequently, various species evolved different types of skeletons to suit their survival needs. In this next section, I'll guide you through understanding the diverse evolution of skeletons in nature. As mentioned earlier, the earliest skeletons likely served as defensive weapons to protect animals from harm. Consequently, exoskeletons enveloping an animal's outer body, providing defense, became the most significant and prevalent form of animal skeletons. Many animals possess exoskeletons, including familiar creatures like mollusks, arthropods, as well as lesser-known ones like onychophorans and tardigrades. Among them, arthropods stand out as the most representative, boasting a vast family that includes millions of species such as insects, crabs, spiders, and the long-extinct trilobites. Assisted by exoskeletons, trilobites emerged as one of the most successful creatures during the Cambrian period, so much so that they have become synonymous with ancient fossilized organisms. Subsequently, arthropods, aided by their exoskeletons, became among the first to transition from oceans to land and from land to the sky. They landed approximately 425 million years ago, about 50 million years earlier than terrestrial vertebrates, and took to the skies roughly 400 million years ago, a whopping 170 million years earlier than flying vertebrates. Presently, insects with exoskeletons remain the most abundant and diverse class of species on Earth. Across all these milestones of evolution, exoskeletons played a paramount role. However, despite their advantages, exoskeletons have a fatal flaw: they limit an animal's size. Although many science fiction works feature giant insect monsters, at least on Earth, exoskeletons cannot support the existence of large-bodied creatures. Why is that? It's an engineering problem. For an animal to grow, its exoskeleton must also grow. How can an animal grow while carrying a shell? Some animals, like mollusks, solve this by continuously growing new shells at the edge of the existing one. However, the largest mollusks only reach lengths of slightly over a meter. Other creatures, like spiders and lobsters, have their bodies entirely encased in exoskeletons and can only increase their size through periodic molting. Yet, during molting, they become vulnerable and prone to attacks. Additionally, as their bodies grow larger, the weight of their exoskeletons increases at a faster pace, easily crushing the animal's internal tissues. Hence, for animals of larger size, exoskeletons are not a viable solution, leading them to opt for endoskeletons. Compared to exoskeletons, the emergence of endoskeletons occurred later and initially represented a non-mainstream skeletal structure, possibly because endoskeletons couldn't provide defense against attacks like exoskeletons. Hence, early endoskeletal animals discovered so far were small in size, possibly a strategy to evade predators. However, in the long run, free from the constraints of an outer shell, endoskeletal animals possessed greater growth potential. Endoskeletal animals constitute a vast family, among which the most prominent are vertebrates. Vertebrates underwent extensive and prolonged innovations with their endoskeletons, beginning with material improvements. Starting from early fish, some evolved an endoskeleton composed of calcium phosphate, distinct from the more common calcium carbonate in exoskeletal animals. Calcium phosphate offers various advantages. It's harder than calcium carbonate, more resistant to acidic seawater, and serves as a reserve of essential elements like phosphorus, crucial for biological functions. In terms of skeletal material, vertebrates took a significant leap forward. Additionally, based on their needs, vertebrates constructed different tissues to create endoskeletons with distinct functionalities. Vertebrate skeletons mainly consist of 4 tissue types. The most common is bone, a living material containing bone cells and bone marrow, crucial for producing blood cells. The second tissue type is cartilage, firm and elastic, predominant in the early skeletal structures of fish and commonly found today, such as in the skeletons of sharks and in human joints. The last 2 tissues are dentine and enamel, which constitute our teeth. The combination of these tissues significantly expanded the functionalities of the endoskeleton. Besides improving materials and adding varieties, vertebrates made numerous structural innovations in their skeletons, gradually seizing control of evolution. One of the earliest major innovations was the appearance of jaws, what we commonly call the lower jaw. Yes, the earliest vertebrates lacked jaws until the early Silurian period, approximately 440 million years ago, when primitive fish finally evolved jaws. This breakthrough endowed vertebrates with the ability to bite, chew, and vocalize, significantly reinforcing their survival advantages when interacting with other species. The subsequent significant innovation was the appearance of limb skeletons. Present-day vertebrates with limbs evolved from a fish called sarcopterygians. These fish had unique fins primarily composed of muscles and bones, quite fleshy compared to the common thin fin-like structures seen in today's fish, resembling miniature arms and legs. Later, some sarcopterygians transitioned from water to land, using these fleshy fins to support their bodies. Limbs evolved from these fleshy fins, and it was with the support of these limb skeletons that vertebrates eventually conquered land. Upon reaching land, taking to the skies was just a matter of time for vertebrates, and during the process of mastering flight, they further enhanced their skeletons. Pterosaurs were the first vertebrates to have active flying ability. Given their large size, these creatures required enormous wings. Their solution was quite creative. While the upper arm bone and the forearm combined accounted for only a third of their forelimb length, the fourth finger bone was exceptionally elongated, supporting the remaining 2/3 of the wing. In some pterosaurs, wings extended to their hind limbs, significantly increasing wing surface area. To reduce flight weight, many of the pterosaurs' bones were hollow. Moreover, to ensure strength, these bones contained numerous pillars and struts, making pterosaur skeletons both strong and lightweight. Later, birds and mammals, like bats, became flight experts, with their skeletal concepts resembling those of pterosaurs. They emphasized lightweight design and modified their upper limb bones to support their wings. Vertebrates exhibited various innovations with their endoskeletons, but due to time constraints, we won't delve into each one. Earlier, we mentioned the definition of skeletons, usually referring to the hard tissues inside or on the surface of animals. Broadening this definition, the author believes that plants and microorganisms also have their own skeletons. For instance, the branches of trees are their own rigid structures that provide support, extending the definition of skeletons. Let's discuss plant skeletons. Today, the tallest trees on Earth can reach over 100 meters in height and weigh several thousand tons. To support such immense structures, sturdy skeletons are necessary. When did plant skeletons first appear? The earliest known plants emerged around 800 million years ago, but it wasn't until 470 million years ago that the first plant skeletons evolved. These plant skeletons existed in the form of tiny spores, extremely tough and serving to protect the spores. The oldest known upward-growing plant was Cooksonia, essentially a branched and photosynthetic stem with a diameter of only about 1 millimeter and a height of a few centimeters. Truly tall trees appeared around 390 million years ago in the mid-Devonian period. Vaiacia, a tree fern-like plant, could grow up to 8 meters tall. By the mid-Carboniferous period, around 300 million years ago, Earth's forests witnessed explosive growth, with numerous unusually shaped, towering trees rapidly emerging and falling. The remains of these skeletons had profound effects on the Earth's climate. What were these impacts? Firstly, the primary component of woody tissues is organic material, which absorbs a significant amount of carbon while growing. Once buried in swamps and deep underground, this sequestered carbon from the atmosphere effectively mitigated the greenhouse effect, lowering atmospheric temperatures and eventually leading to the formation of extensive ice sheets in polar regions far from the tropics. Secondly, as plants competed for sunlight, they grew taller, inadvertently providing a more diverse habitat for animals, indirectly promoting animal evolution. Plant roots penetrated deeply into the ground, accumulating thick layers of soil and altering the flow of surface rivers. Finally, these plant skeletons, buried deep underground, turned into today's coal, coincidentally becoming an essential fuel driving human technological advancement. Clearly, plant skeletons had a profound and far-reaching impact on future generations. Apart from animals and plants, microorganisms also possess skeletons, with a much longer history. Scientists extracted extremely ancient microbial fossils from rocks dating back 3.2 billion years. These microbes had small spherical bodies surrounded by complex, tough organic shells, possibly the oldest microbial skeletons on Earth. Some theories suggest that these might have been structures for self-protection in harsh environments. In the subsequent evolutionary process, microbial skeletons displayed considerable creativity in material selection. For instance, foraminifera, commonly found in oceans, built their skeletons from organic material they produced themselves. At times, they embedded sand grains into these organic structures, while others extracted calcium carbonate from seawater to construct their skeletons. Moreover, some microorganisms used silicon dioxide, the primary component of glass, to create their skeletons, the most notable being diatoms. Diatoms are crucial marine producers, providing nearly half of the basic food supply in the oceans. Despite their tiny size, diatom silica skeletons emit dazzling iridescent colors under a microscope, considered artworks in the microbial world. Part 3, Earth's life forms have always been brimming with creativity. Animals, plants, and microorganisms have evolved an array of skeletons according to their needs. These skeletons are not just vital for survival but also repositories of valuable scientific information. In this final section, let's explore the secrets unearthed through skeletons. Isotope analysis is a crucial tool in paleobiology. By examining isotopic contents within fossils, we glean invaluable insights. For instance, analyzing oxygen isotopes helps reconstruct temperature changes in ancient eras. There are 3 oxygen isotopes in nature: oxygen-16, oxygen-17, and oxygen-18. The ratio between them relates to environmental temperature. Oxygen-17 is typically disregarded, so by detecting the ratio of oxygen-16 to oxygen-18 in fossils, we can deduce the environmental temperatures of ancient life. In the Cambrian and Ordovician rock layers, fossils called conodonts are abundant. Analysis of oxygen isotopes in these conodonts revealed that during the Cambrian, tropical ocean surface temperatures soared to 40 degrees Celsius. In contrast, today's tropical ocean temperatures are around 30 degrees Celsius. This prolonged high temperature persisted for millions of years until gradually cooling to levels akin to today's during the mid-Ordovician. How does one explain such temperature variations? Fossils provide a potential answer. Scientists observed a rapid increase in ancient biogenic skeletons within marine sedimentary rocks starting from the Ordovician. This proliferation likely sequestered carbon, reducing atmospheric carbon dioxide levels, lessening the greenhouse effect, and eventually cooling the Earth to a relatively suitable temperature. Apart from tracing temperatures, fossils enable exploration of past time spans. As Earth's rotation speed diminishes over time, what was the length of a year in ancient times? Animal skeletons also preserve this data. Tidal seasonal changes impact marine life growth. By examining their skeletal fossils, we can reconstruct ancient tidal patterns and the length of months and years. Analysis of a 360-million-year-old fossilized coral revealed that a month at that time spanned 30.6 days, with a year comprising 13 lunar months, totaling around 398 days, markedly different from today's count of 365 days. Zooming into more recent history, analysis of animal skeletons from the past few hundred years allows accurate reconstruction of short-term climate changes. In 1868, a long-lived ocean quahog captured in Icelandic waters was estimated to have been born around 1494, living through more than 300 years. Through analysis, it revealed significant details. For instance, during this quahog's 300-plus years, particularly between 1816 and 1818, its growth rate drastically slowed. This period coincided with the year without a summer, in 1816, attributed to the severe sunlight blockage caused by the eruption of an Indonesian volcano, resulting in the coldest year in the past 500 years. This correlated remarkably with the quahog's growth pattern. These concealed details within quahog skeletons are vital, providing records extending before the 15th century when scientific climate recording wasn't prevalent. Uncovering this missing climate data in animal skeletons greatly enriches our environmental observation records and enhances our climate prediction capabilities. Animal skeletons unveil nature's mysteries, while human skeletons aid in reconstructing historical truths, revealing many details absent from written history. For instance, in 2012, British archaeologists discovered the long-lost remains of England's last Plantagenet king, Richard III, beneath a parking lot. Through thorough analysis, scientists nearly reconstructed Richard III's entire life. How was this achieved? Isotope analysis played a significant role. Combining different characteristics of various bones, isotope analysis extracted many details. For example, dental enamel completes its development during childhood. Analyzing isotopes in enamel helps trace a person's growth history. Strontium isotopes indicate a region's geographical location, while oxygen isotopes reveal rainfall patterns. Isotopic analysis of Richard III's dental enamel indicated that he relocated from Northampton, his birthplace, to a Rainier location near the Welsh border at age 7. Similarly, analyzing bones that continued to grow after childhood, like isotopes within toothed dentin, helps reconstruct later living environments. Results showed Richard III moved to a drier area in his later years. Lead elements in his teeth suggested air and soil quality had deteriorated during his time. Finally, because ribs continuously repair throughout life, analyzing carbon and nitrogen isotopes within ribs allows reconstructing the lifestyle of the skeleton's owner. Researchers found Richard III had a varied diet, enjoying wildfowl, freshwater fish, and red wine. Richard III probably never imagined that his life would be faithfully recorded by his own bones and presented to later generations in this manner. Here, the essence of this book has been covered. Let's summarize briefly. We first discussed the origins of skeletons and why living beings evolved them. The oldest known skeletal fossil, the Claudius fossil, dates back 550 million years. From the Cambrian explosion around 540 million years ago, the number of species with skeletons increased rapidly. Scientists suggest that skeletons might have initially served as defensive weapons for protection. We then explored the diverse evolution of skeletons in nature. Animal skeletons can be classified as exoskeletons and endoskeletons. Exoskeletons appeared earlier, with over 1 million species, albeit generally smaller. Endoskeletons later emerged, undergoing material, functional, and structural innovations in vertebrates, becoming a cornerstone of advanced animal evolution. While plant and microbial skeletons do not fit traditional definitions, they are equally significant. Plant skeletons dramatically altered the Earth's ecological environment, leaving behind valuable fossil fuels. Microbial skeletons preserved the oldest traces of life, crafting the most vivid microcosms. Lastly, we delved into the past secrets unearthed by skeletons. Analytical methods such as isotope analysis extract rich data stored in bones, revealing natural shifts in temperature, the duration of months and years, natural disasters, historical events, making skeletons true witnesses and honest recorders of history. The author highlighted a startling figure: a mere 5% of the total weight of wild vertebrates on land accounts for less than human and artificially bred animal masses. This indicates extreme marginalization of wildlife, with humans dominating land vertebrate skeletons. Moreover, human alterations to skeletons are immense. Selective breeding has created vastly different skeletal forms in dog breeds, nearly unrecognizable as the same species. Fast-growing broiler chickens raised for just 6 weeks exhibit rapid but compromised skeletal growth compared to their chicken ancestors. It seems humans have assumed dominance in shaping skeletons from nature, yet can we responsibly use this power for our and nature's benefit? Perhaps, this is a question nature leaves for humanity. That concludes the essence of this book. Congratulations on completing another book. Thank you for your support and attention. Subscribe to the Tim Booker channel, like, and share this valuable knowledge with friends around you. Let's combine wisdom with action, achieve our financial goals, and create a better future together. Thank you all, goodbye.